So the topic is sources of drugs. So by sources of drugs, we simply means that the sources from where the drugs are obtained. Like there are natural sources, and there are semi-synthetic drugs, and there are synthetic drugs. So natural sources mean they are present naturally in the environment, and we make drugs out of them. So examples are plants, then animals. We have others like microorganisms and minerals. So all these are the natural sources present in our environment, and they can be important sources of drugs. Then examples of drugs that are derived from plants are morphine, atropine, quinine, and digoxin. So morphine, a common painkiller; atropine, an anticholinergic drug used for symptomatic bradycardia; quinine, an anti-malarial drug; digoxin, used for improving the heart function in heart failure. So all these drugs are plant-based drugs. Then animal sources drug include insulin and heparin. We know heparin is a common anticoagulant, and insulin is used for treating diabetes. We know minerals sources drugs include ferrous sulfate and magnesium sulfate. Both contain minerals. Then we have microorganisms based drug. Penicillin, an important antibiotic, is derived from a microorganism source. Then we have another class of drug which is known as semi-synthetic drugs. So semi-synthetic mean they are obtained from the natural sources, like for example they will be obtained from plant or animal source, and then they will be later modified into the labs. So that is semi-synthetic. They are known as semi-synthetic. Semi mean half synthetic, because they are obtained from the natural sources and then they are modified. So examples of such drugs are hydromorphone and hydrocodone. Again, a hydromorphone, a common painkiller. So that's a modified form of morphine. That's why it's known as hydromorphone. Then we have synthetic drugs. So synthetic drugs are totally artificially produced. Okay, so they are not obtained from the natural environment. They will be totally artificially produced. Therefore, they are known as totally synthetic drugs or synthetic drugs, and they are produced artificially. Most of the drugs that we use nowadays are synthetic drugs. Remember, like paracetamol or any drugs of the companies like aspirin, paracetamol. All of them are synthetic drugs. So in today's world, majority of the drugs are synthetic. Then we have another concept, and that is genetic engineering. So what is genetic engineering? This is a new concept related to DNA recombinant technology. And genetic engineering can help produce a large number of drugs. For example, human insulin, human growth hormone, hepatitis B vaccine. So these are examples of a number of drugs that can be produced by genetic engineering technology. Basically, genetic engineering technology involves taking a small gene from the human, which will be encoding, for example, insulin or a growth hormone, and we will implant this cut gene from the humans into the bacteria or other organism, and that will produce the proteins, uh, for example insulin and growth hormone, that are needed. To better understand, look in the figure. On the right side we have a human cell containing DNA, and this DNA, for example, will be encoding for insulin. So we will take up the DNA or gene from the human cell. And on the left side we have a bacterial DNA. So bacterial plasmid or DNA, we will cut it, and we will implant the part of DNA of human which is encoding for insulin. That will be implanted into the DNA of the bacterium. And now the modified DNA of the bacterium, which now include the insulin encoding gene which we have taken from the human cell. So our aim is to produce insulin. So insulin producing gene from the human cells is implanted into the DNA of the bacterium, and this is now implanted into the bacteria. Again, the gene is implanted, the whole DNA with the modified gene from the humans encoding insulin is implanted into the bacteria. Now when bacteria multiply, the gene will also multiply, and it will produce the protein, which is our resultant insulin. So this is how a large number of recombinant insulin can be produced with the help of genetic engineering technology. So this is an example of how genetic engineering technology works. So it is very simple that you take up a part of gene from a human cell which is encoding, for example, insulin or a growth hormone, and you will implant it into the DNA of the another organism, for example, bacteria. And when the bacteria multiply, it will express our implanted gene, and the resultant product, for example insulin, as the gene express, we know it will produce proteins, and these proteins will be making our insulin or growth hormone. So that is the basic concept of genetic engineering technology.